Hi guys, welcome back to my channel. So today's video, as promised, is a B1 summary for the exam board OCR 21st century, not the new spec, old spec, biology first module. And before I start this video, I do want to apologise because sometimes my paper does go off screen slightly, and that's because it's my first time filming this type of video and the camera positioning was slightly off. So with that being said, I do hope you guys find this useful and helpful, and if it does, please like, comment and subscribe, and yeah, share it to anyone doing the exam board. So yeah, let's go.
B1. You and your genes. This is an animal cell. An animal cell contains a nucleus, and inside that nucleus you will find chromosomes. Exactly 46, i.e. 23 pairs of chromosomes. Inside a chromosome you'll find DNA. A short section of the DNA is a gene. A gene carries instructions that control how you develop and function. They code for proteins. There are two types of proteins. The first type is a structural protein. These give the body structure, rigidity and strength. For example, hair, nails, blood, and an example of one is collagen. The second one is a functional protein. This enables the body to function, for example, enzymes or antibodies. And an example of an enzyme is Alleles. A human egg cell has 23 chromosomes. Not 23 pairs and 46 chromosomes, but only 23 chromosomes. The same as with a human sperm cell. When the egg cell and the sperm cell come together during the process of fertilization, they form a newly fertilized egg, i.e. a zygote, which contains 46 chromosomes, i.e. 23 pairs. This means that 23 of your chromosomes come from your dad and 23 from your mum, making you have 46. The differences between individuals of the same species are described as variations. Variations may be due to the genotype or the phenotype. The genotype is the genetic makeup of an organism, whereas the phenotype is the observable characteristics the organism has. The environment can change a phenotype. Identical twins have the same genotype, but any differences in the way they look is due to their phenotype, and that is a result of their environment. Non-identical twins, on the other hand, have different genotypes and different phenotypes. This is due to the way they both have been conceived. Identical twins have come from the same egg and same sperm cell, whereas non-identical twins come from different sperm cells and different egg cells, just like other siblings. And also remember, a boy and a girl can never be identical twins. I was pretty in denial in year 7 and even decided to have an argument with my science teacher. Now, parents pass on their genes to their offsprings in their sex cells. A pair of chromosomes carry the same genes in the same place on each chromosome within the pair. However, there are different versions of a gene called alleles. These alleles may be the same or may be different. Same alleles are known as homozygous and different alleles are known as heterozygous. The chromosome we get from each pair is completely random, and that is why every child in every family looks similar but not the same. The child will share similarities with its parents, which are dependent on their mother's alleles, their father's alleles, and whether those alleles are dominant or recessive. If an individual has one dominant and one recessive allele, they will show the dominant trait. If an individual has two dominant alleles, they will show the dominant trait. Whereas if they have two recessives, they will show the recessive trait. In the exam, you will need to know how to interpret genetic diagrams. These are called Punnett squares, and this one is a family tree genetic diagram. Now, for the Punnett squares, you put the mother's alleles and the father's alleles on one side and the other. Then you use it like a multiplication grid and you work out the possible combinations of alleles you can get. Now, the first two I have down is heterozygous dominant and the other two are homozygous recessive. So the first will show the dominant trait and the second will show the recessive trait. And a child can never be heterozygous recessive because the dominant allele will always overrule the recessive allele. Now I'm doing the same on the other side but with different letters, and you'll be asked to work out the probability of these. And the family tree, you should be able to interpret these in the exam. And you can get 6 markers on them. The first genetic disease you need to know about is cystic fibrosis. This is caused by a recessive allele. Now, with recessive allele diseases, you can have people that are carriers of the disease, which means they will have a recessive allele and a dominant allele. But because of the dominant allele, they would not have the condition. Therefore, the chances of this family in the corner here having a child with cystic fibrosis is 25%. Carriers will not inherit the disease, but they are capable of passing it on. Now, as well as knowing the probability of finding the symptoms of cystic fibrosis include a thick and sticky mucus, breathing problems, and difficulty digesting foods. Also, you will find that people with cystic fibrosis get severe chest infections, and there is no cure, and it is not contagious as it is a genetic disease, which means you cannot catch it. It's also important to note that the thick and sticky mucus is found in the gut and pancreas. Now, Huntington's disease is the other genetic disease you should know about, and this is caused by a dominant allele. Now, if a mother doesn't have Huntington's disease and the father does, this is how you would draw the Punnett square for one. Now, the dad only has one dominant allele, but even if he had 1 or 2, he'd still get Huntington's disease. So the chances of them having kids with Huntington's disease is 50%. And there can never be carriers as it is caused by a dominant allele. Now, the symptoms of Huntington's disease include uncontrollable shaking, clumsiness, memory loss, inability to concentrate, and also mood changes. Now, for the exam, you would also need to know about genetic testing. This is prenatal genetic testing. There are 2 types of methods for prenatal genetic testing, and one is amniocentesis and the other is chorionic villus sampling. Prenatal means it is done before the baby is born. Now, with chorionic villus sampling, there is a 2% miscarriage risk and results come at 10 to 12 weeks. This can also be seen as a positive though. However, there is almost no risk of infection. But on the other hand, it is also worth considering that the results are not 100% reliable. Now, with amniocentesis, there is only a 1% miscarriage rate. However, there is a small risk of infection, and the results come at 15 to 18 weeks, which is later than the results for chorionic villus sampling. And it is also not 100% reliable. Now, with genetic testing, it can be done on adults or even babies like we did before. There are different types of methods, and one includes having a DNA kit that can tell if any faulty alleles are present. It is done to know if a genetic disorder runs in the family. It can be done by both adults and babies, and it is done on individuals. However, genetic screening is done on the whole population. All babies in the UK are scanned for cystic fibrosis. It costs money for the NHS to do it, but they do it for free, whereas I think genetic testing you'd have to pay privately. Now, genetic testing and genetic screening helps changes lifestyle and makes important decisions and allows you to plan ahead. Now, in this topic, there are ethics regarding genetic testing and screening. One is that it's very dangerous and people shouldn't worry about life in the future and they should just live life now. And another person would say, don't interfere with nature. Another person would say, we should interfere so that we could plan ahead in case anything is wrong with the child. And another person is saying, if my husband and I both had 40 alleles, we'd be upset/scared about having kids. Now, after genetic testing is done, potential parents can pick out the embryos they want, the embryos that do not have the 40 alleles, and choose to follow on with the IVF process if they wish to do so, which is also known as in vitro fertilization. Now, one of the other ethical matters with genetic testing and screening is the potential to have wrong results. So, for example, false negatives and false positives. This table shows what each of them are. Clones are genetically identical individuals. Bacteria, plants, and even some animals can reproduce asexually to form clones that are genetically identical to their parent. Asexual reproduction only requires one parent, unlike sexual reproduction, which needs two. Since there is only one parent, there is no fusion of gametes, i.e. the sex cells, and no mixing of genetic information. As a result, they are genetically identical to their parent and to each other. Identical human twins are known as naturally occurring clones. Plants reproduce asexually, and they can take place in a number of forms. Some plants, such as strawberries, produce shoots called runners. These eventually break off and become new strawberry plants, which are clones of the original. Other plants grow bulbs. When bulbs are planted, they grow into genetically identical plants. Again, the environment will alter them. Now, animal clones can occur naturally and artificially. Artificial cloning is the process by which the clone Dolly the sheep was created. Now, this diagram illustrates how the cloning process takes place. The sheep that is born is actually a clone of the white-faced sheep, as it contains the same genetic information as the white-faced sheep. The black-faced sheep at first just gave a donor egg, the nucleus was removed, and the third sheep was just a surrogate mother. Cloning depends on cells that have the potential to become any cell type in the body. These are called stem cells. Adult stem cells are unspecialized cells that can develop into many, but not all types of cell. This is illustrated by the diagram seen on the screen. Now, embryonic stem cells are unspecialized stem cells that can develop into any type of cell, including more embryonic stem cells. As a result of being unspecialized, stem cells from embryos and adults offer the potential to treat some illnesses. For example, skin can grow as a treatment for serious burns, and sight can now be restored to people who are blind due to damage of their corneas. The majority of cells of multicellular organisms become specialized during the early development of the organism. This is because after the zygote has divided 4 times to reach the 16-cell stage, the majority of cells in the embryo start to become specialized. This is later repeated in B5 stem cells, so that is something you would have to expand on in B5. So that was a quick summary on B1, You and Your Genes. I hope you guys found it useful, and stay tuned for next week's video, which will be uploaded on Monday.